So we're going to hear from the next presenter, who is no other person than Yuval Dorfan, who is founding director of the Israeli SynBio Institute. Yuval is the founding director of the Israeli Innovation Center, where he leads integrative and applied research at the IDC, also involved in generating or raising funds to enable the kind of work that is done at the IDC, and also working collaboratively with people in industry and also academics to conduct research, and not just any research, holistic research, to enable the creation of new companies. So we will then welcome Dr. Yuval Dorfan, who would speak to how his work is enabling the creation of bio ventures. Thank you. Apologies for the delays. We are trying to get the speaker to present his slides. If not, we may be hearing from another speaker. So while we try to get in touch with Dr. Yuval, we would transition to hearing from Dr. Jenny Molloy.
Excuse me.
Right.
Okay. I hope you can hear me also.
Loud and clear.
Excuse me?
Yes, we can hear you loud and clear.
Okay. So I'll start. Okay, so thank you for the invitation. It's a really honor to speak here. And we also collaborate with lots of different groups around the world. And I'm happy to speak about the soil water biosensors application driven by a new Israeli bio-foundry that we're starting now called Allergen. I'm from the Reichman University in Israel, and this work is a collaboration with the Hebrew University, MIT Broad Foundry, where I was a research scientist till 2020. Together with High Labs, we're now ramping up Allergen. And another collaborator is Bar-Ilan University from the Nano Center from Bar-Ilan University. Okay, so I'll speak about a few applications for biosensors. Then I will focus mainly on old landmine detection, and I will show how application-oriented research can be performed. As you know, Africa is one of the places where humanitarian landmine is a very big issue. I will share a few initial results using a technology called mini-ice, and then show you how the new foundry can accelerate such a project like this. So as you know, there are lots of biosensors based on microbial or other organisms. I'm focusing here on three types of applications. The first one is air quality. So we have, together with the Nano Center in Bar-Ilan University, we have a project for air quality. We're looking there with a biochip for three different types of contaminations: viruses, bacteria, and small molecules. Another family of applications is water quality and biosensors for water. And I'm going to focus on work that was started many years ago by Professor Shimshon Belkin and other partners from the Hebrew University for the landmine detection. So, as you probably know, it's a global problem. We have very large areas where we know that there are landmines, but we don't know where they are. And as I said, Africa is one of the most severe places where we need to solve the problem. The problem is that the technology of metal detection is obsolete because the landmines are plastic, wrapping some explosives. So the only way we can detect them is based on the diffusion of contamination into the soil. So I will show you how we're focusing on this project with synthetic biology. So just before that, mentioning what was done already based on E. coli. Professor Belkin took the model organism E. coli and discovered that it can detect DNT, which is very common in Israel because the landmines contain TNT. And as you can see here, the detection is very accurate. They spread the bacteria, and after a few hours, they scan the area optically and get very good signals from the areas where there are landmines. But this works in certain conditions, and there are issues to be resolved. One thing is the robustness to environmental conditions. And the other thing is that this solution is quite expensive because E. coli is not a good candidate to survive in dry and extreme temperatures as we can find in a typical landmine field. So together with them, we plan to switch from the model organism to native strain. And actually, it's a way of thinking. Since we're in synthetic biology, we're application oriented, we start from a sampling of soil in this case, unlike starting from a model organism that we can work in the lab, and the lab is very equipped for that. We start from a sampling of relevant soil. The motivation is what I said before, robustness to environmental conditions, but also if we get samples from old landmines fields, probably the bacteria already know that there are some landmines there because DNT is an energy-rich molecule. So our assumption is that we will find native strains that contain already the sensors. They know that they already sense it. They didn't tell us that the landmines are there. So we will add the reporter to them, and we can use those bacteria either with known sensors that are already published, or we can also discover new sensors. So we adapt our lab to the application, and then we have an intermediate step between the lab and the field before we get to a proof of concept to be able to screen for various conditions before we get to expensive field trial. We work with the agile spirit. It means that we can iterate from each step to another until we get and converge to a proof of concept. One of the techniques that we're using is mini ice. Ice is a natural phenomenon that a donor strain transferred DNA to another bacteria called recipients. We base our research on the mini ice. It's a technology developed by Christopher Voigt in MIT, and I will show some initial results with this technology. So as I said, we start from samples, in this case various geographical areas, soil, mud, and water, and what we got is that for any of those samples, we were able very quickly with very simple protocol to engineer many strains. I'm showing here some of those strains. And we were able to engineer them and get a very strong GFP signal. and we were very happy with the results. And not surprising, but very encouraging, is that those strains, these are the closest relatives of the strains we engineered from NCBI. So what you can see is that they're very robust to extreme conditions like high temperature, high radiation, etc. Last thing I want to cover is how we're going to build allergen, the bio-foundry, and I will use this example, this project, to show how it can accelerate such projects. But of course, the company is going to support any kind of project in synthetic biology. And the vision is that we can take any idea from industry or academia and get the project to a proof of concept as quickly as possible. So you probably know the design, build, test cycle. We changed it a bit. So we start from computational design, then we switch to automated build. In this case, many strains and many sensors and the combinations between them. Then we get to high throughput screening within the lab. But then we add a fourth step, which gets the project to a much more mature step. We call it scale. And it means scale out, getting out of the lab, showing that indeed we can detect landmines in the field and not just in the laboratory, and other aspects of getting out of the lab. And scale up. Scale up means producing lots of bacteria. We have big areas, we need lots of bacteria. In other projects like bioproduction, scale up means fermentation, means producing more and more of the product in an efficient way. If we succeed in the fourth step, we finish the project. If not, we can iterate to any of the steps back until we get to the proof of concept. So that will conclude my talk. I just want to thank my group that is growing very quickly. My university, Reichman University, the Hebrew University, MIT Broad Foundry, where I did my postdoc, and Bar-Ilan Nano Center. And last but not least, High Labs, a company in Israel that we partnered with them. They are a service company in biotech, and together with them, we ramp up now allergy, the Israeli biofoundry. Thank you.